I was looking at the Thomas PET scan, where one's functional brain was silenced by trauma. Besides Tom, we also had 56 other individuals in the study. 17 of them were previously convicted, 20 of them were involved in some type of accident which caused them to have a traumatic brain injury, and 20 of them were normal controls, and we used their brain data to compare with those two specific population of patients. And the ultimate goal of the research was to try to understand what specific brain regions were impaired in these two population of patients, and most importantly, how we can restore the pathology after identifying the brain regions. Because violent behavior has been shown to be linked to neural dysfunction, which means that there are specific brain regions that commonly show up upregulated or downregulated in this population of patients. So the results of the study showed that people who were previously convicted, they had hyperactivity in the parts of their brain responsible for aggression, and that was limbic region and cortical regions. And we also had hypoactivity in the parts of the brain responsible for decision making and impulse control, which were prefrontal cortex and other brain regions involved. However, in patient population who were impacted by accidents and had traumatic brain injury, we saw that they have compensatory activity in frontal and temporal brain regions, which were parts of the brain involved in emotional regulation and decision making. Specifically, that it was anterior cingulate cortex, medial prefrontal cortex, and other brain regions. And now, after knowing this brain region specifically, if we can find ways to reverse the pathology by neurostimulation, by cognitive training, or by metabolic intervention, then we can potentially improve their brain health, and doing that will prevent future violence from happening. Or if we can able to identify these markers early on in specific patients, we can prevent future violence from happening. So changing the brain can change the future of these patients, and by understanding the specific brain regions impaired in this population of patients and the ways we can restore their metabolic function, can bring the science into real world impact and contribute to lives of these patients and future.